Hi, thanks. Thank you again to the organisers for organising this conference and also letting us talk about the technology that we have at BitBio. I'm currently a team lead in the cell development department, and my main focus is our immune-based programs that we have running. So we are quite aware in the audience already of the cell problem when we're looking at cells to use as in vitro models for drug development, disease modeling, and if we look at in vivo models, it doesn't always recapitulate, well, most of the time it doesn't recapitulate human disease, which makes it, adds another bottleneck to the development of small molecules, compounds, and also therapeutic antibodies for therapeutic approaches. And there's also a shared problem which also comes across with the emergence of cell therapies as well. And that is an access to reliable human cells which are scalable and consistent. And these mainly come under the banners of we need better methodologies to manufacture human cells, one for research and development and drug discovery, and the other for the potential of cell therapeutics. But we're faced with sort of bottlenecks of scalability. With iPSC-derived therapeutics, we have the issues of prolonged culture to generate the cell type that we're after. We have issues of purity, and we have also issues with inconsistency. So we focus on generating cell types from induced pluripotent stem cells at BitBio, and we focus on a wide range of cell types within the body. And to generate iPSC-derived mature cell types, there are a couple of methods by which we can do this. The first one, which has already been touched on today in generating iPSC-derived microglia, is using the directed differentiation approach, where you recapitulate the developmental process, but within a dish, using media compositions, growth factors. And this can be expensive because you have to do various different stages of different media cocktails. And this can take up to 20 days, 30 days, 40 days, depending on what cell type that you're trying to generate. So this in itself brings issues with purity, because you will get non-cell types of interest present during that differentiation process, which then will impact your scalability. And again, it's a long process. It takes a long time. It's hands-on. It's cost. If you've got something that's long and an infection happens, that's everything down the drain. There are other processes by which you can generate iPSC-derived cells, and this is using something known as forward cellular reprogramming, where you take the sort of code for the cell type that you're trying to make. So we use transcription factors in this instance. You plug those transcription factors into the iPSCs and hope that those transcription factors can drive that process from iPSC to mature cell type quicker and give you a more defined cell type in the end than you do with the direct differentiation. So this helps with the purity because you're putting in the code for what you want to generate. And then this will translate to scalability because the hope is what you're putting into your cells, you should get a homogeneous population. However, depending on your targeting approaches, you could get silencing of what you're putting in, which then could impact your scalability. But it does also have a benefit of shortening that culture period point as well. So what we have at BitBio is a sort of three-pillar platform, so a cell identity coding platform. We start off with a discovery platform where we use a mixture of bioinformatics tools and culture to identify transcription factors that can take us from the cell type that we would like to make using an iPSC and identifying those transcription factors in that cell type that we're after. And then once we've identified our transcription factors, we take these transcription factors and plug them into our Optiox technology. And then we do some further fine-tuning of culture conditions because, of course, culture condition and environment is also important to maintaining cell identity. But how does this help? Well, by doing this, we get a greater precision over what we need to generate. This also helps with the scalability. So with our system, we don't see silencing that you may see with other methods to deliver transcription factor-specific reprogramming. And then, of course, we have the benefit of a shorter culture period time. So what is Optiox and how does it overcome the silencing that we might see? So with our approach, we have a tighter control of the expression of the transcription factors, and this is by targeting to multiple genomic safe harbors. And just to exemplify this, we have GFP here. which when the GFP is switched on, you can see a nice homogeneous expression within the cell population. This is just iPSCs. And then when GFP is off with a TEC-inducible system, you do not see any GFP expression. So this helps to overcome that silencing that you could see with other transcription factor delivery methods. And here is just an example of our cardiomyocytes that are differentiating the dish, so from the iPSC stage all the way till the end, and you can see you get a really nice homogeneous reprogramming response. And we hope to use these cells for R&D purposes, drug screening, antibody screening, also cell therapeutics, and also biomanufacturing processes. But as we are at a Microglia conference, I would like to talk to you about one of our first immune cell types that we have recently released, and that is our IO microglia. So let's just get the video going. So the microglia that we have, they come as cryopreserved. We do a pre-reprogramming step in-house from iPSCs, and then we have a cryopreservation point. So if you were using this or were to use this as a customer, these are for R&D purposes only for now, it takes about 10 days after thawing, and then you have your microglia in the dish. And what this video is showing is just an InQSight video of how the cells look from thaw all the way to when they are ready for functional assays and characterization at day 10. You can take them a little bit earlier, but our most optimized time point for functional assessments has been day 10 with these cells. So to discuss a little bit about the purity, we always characterize our cells with flow cytometry. And you can see that you get a nice purity, especially for your CD14, which is your sort of myeloid lineage marker, and your CD45, your pan-hematopoietic pan-immune. You get a really nice purity of, in this instance, it's 98.7% and above. So the purity is there, which is great, along with the key markers that you would expect to see in microglia, so TMEM119, P2RY12, and of course CD11B, which is your pan-macrophage marker. We also confirm whether we see any contamination of pluripotency, so any stem cells left over. And between the batches that we have generated so far, we don't see any markers of pluripotency when compared to our iPSC starting material control. So we can see lot-to-lot consistency with our phenotype and also with the presence or absence of any contaminating cell types. And this is also further exemplified with the morphology when we look at ICC and markers via ICC expression, and also from some of our external testers and collaborators. So here is just some internal data where we've looked at IBA1 on the confocal microscope, which I quite like this image, and also TREM2 and P2RY12. And so you can see you've got the slightly ramified morphology, but not as nice as Jenny's ramified morphology that you showed earlier. But homogeneous expression, again, of those markers that we're after. And with one of our customers here's some external data just showing the reproducibility, not just in our hands, of CX through CL1, TREM2, and P2RY12 expression, and that nice morphology as well. But of course, function is key for these cells, like any immune cell. And we have some really nice functional data that we have achieved with these cells. So we've got a NanoLive video here, and this is in collaboration with NanoLive. And what is nice is you can see the phagosome. I wonder if I can. So if you just look in the corner here, you can see that these are Frodo-label particles, so that when they're taken up in the phagosome, they don't fluoresce until they've been degraded. They don't fluoresce until that pH has dropped. And what's nice about the NanoLive is that you can see them being taken up and then fluorescing within the phagosomes if you just focus in that bottom right-hand corner, which is very cool. But we've also, as part of our QC, we do look at phagocytosis because we want to make sure that the functionality is consistent between each batch of cells that we make. So with the same E. coli particles here, we've shown that we get lot-to-lot consistency with Frodo-labeled E. coli particles. This is looking at, we do a quantification, so we look at percentage of the cells within the dish that are phagocytosing. We also look at intensity of phagocytosis as well to make sure that's the same. But we also have external customer data as well here that shows Frodo-labeled bacterial particles, so S. aureus here, amyloid beta and latex beads, you see a nice homogeneous level of phagocytosis with the different stimuli in customers' hands. And if you play around with pre-treatments to inhibit the inflammatory response before putting on those particles, or anti-inflammatory response, you do see differences in responses. So for instance, if we look at the pre-treatment with IL-10 and dexamethasone, which should have a negative impact of the pro-inflammatory response, but would enhance phagocytosis, you see an increase in phagocytosis in comparison to the control with that pre-treatment. So that's quite nice to say that if you want to look at the effect of different compounds and antibodies under different stimuli, if you want to play around with the anti and pro-inflammatory responses of these cells, you can do this, and this has been done. But on top of that, we look at cytokine secretion as well. So this is just internal data sets to show that when we stimulate our cells with LPS and interferon gamma or E. coli particles, we get that pro-inflammatory response, so secretion of TNF-alpha, secretion of IL-6, and this is reproducible between each batch that we generate. And external data that has been generated by Eve Corrie and Emma Jones at Medicine's Discovery Catapult have also shown similar responses. You stimulate the cells with LPS, you get this pro-inflammatory response of the TNF-alpha, IL-6, IL-1 beta, but they've also shown that our cells are capable of secreting chemokines as well, such as the CCL2 and CCL3. But in addition to that, they have done some further work for us, where they've shown that our iron microglia are capable of chemotaxis. So they've done this with C5a, where the C5a is in the reservoir here, the cells are within the vehicle here, and they see a dose response in terms of the microglia migrating through the insert into the reservoir with an optimum concentration of C5a at 10 nanomolar. So our cells are capable of chemotaxis as well as the typical phagocytosis and pro-inflammatory cytokine production. But not only that, to try and look in disease states, you want to be able to culture these with multiple different cell types as well to try and get a better recapitulation. So internally, we have a quite large catalog of neurons. We have our ioglutamatergic, GABA, we've got some oligodendrolyte cells, and we're also working on astrocytes at the moment. But we can co-culture our iron microglia with our glutamatergic neurons, either 24 hours post-thaw, or when you get them to that day 10 stage, you just drop the microglia on top of the glutamatergic neurons. And this is the morphology that you get after 8 days in culture, which is a very nice ramified morphology. And you can see that those ramifications are sort of, they look like they're interacting with the neurons. The yellow there is the IBA1 for the microglia, and then the red, the MAP2 for the neurons. And just to see what they look like, you can see them moving around in the dish. This is just on the incus. Oh, this is a little bit blurry, but they look, you should be able to see the microglia sort of wiggling around in there, interacting with the neurons. Doesn't work as I expected that would be, but you can sort of see them moving around. It's better if it's sped up, which means that these cells are co-culture compatible. But we will be working on tricultures with the other cell types that we have in development as well. But another thing that we ensure that we do on all of the products that we make are sequencing. So we do both bulk RNA-seq and also single cell. And then this is just to highlight the consistency between the batches, because this consistency is key. This is just exemplifying a PCA plot of iPSCs before we reprogram the cells, at day 1, which is straight out of the vial, and at day 10. And you can see that between the 3 lots, you get a nice lot-to-lot consistency. And also the single cell data as well helps exemplify this. So this will be 3 independent runs of 3 different stages of the reprogramming phase, iPSC at the beginning, a mid-stage here, and then obviously the microglia at the end. And you see enrichment of the key microglia genes mainly within this cluster here, and everything is quite homogeneous in the process. And when it comes to benchmarking, yes, it is difficult to get hold of primary cells to benchmark these cells to, so we have to rely on external data sets. So we have looked at comparisons between our cell types and published data sets, one of the protocols being the ABOD protocol. And our microglia do cluster relatively closely when looking at differential gene expression to the ABOD et al. Microglia, which are here, but also the fetal microglia and also the adult microglia that are also from that same paper. So in terms of benchmarking against available data sets in microglia, they do cluster quite similarly. But also Charles River has recently presented a poster at the Alzheimer's Association meeting this year, looking at similarities between stimulation of primary human microglia and our iron microglia. And with the treatment of LPS and dexamethasone, they looked at TNF-alpha secretion and IL-6, and you can see that both cell types respond to that stimulus in differing levels, but they do both respond in a similar way. And when we looked at, when they looked at the ability for the microglia to produce inflammasome activation with nigericin, you can see that both the primary microglia and the iron microglia respond and produce that inflammasome, and you can inhibit that inflammasome in a dose-dependent manner with an NLRP3 inhibitor, showing that these cells can be used for drug screening, antibody screening, and compound screens. So just to summarize, at BitBio, our aim is to generate a scalable source of iPSCs. So with the microglia, we can generate billions of cells per batch. So each batch represents a billion cells, with a nice level of consistency, but not only in phenotype, but in function as well. And we make sure that we do a functional QC on every batch that we make. We have defined, because we can see this specific key markers of choice. We see this in the sequencing data as well. And you have usable cells within 10 days, which does help a lot when you compare it to the much longer protocols. We are also looking at disease models for the microglia, so we do have 2 disease models in the pipeline, as well as the other cell types that we generate. So on that note, I would like to say thank you, but the slide's not changing. But thank you for inviting us again and listening to the talk. There is an image of everyone in the team. So just imagine that's the Babraham campus with a load of people standing in front of it. Any questions?
Any questions? Up back.
Hi. You might not want to answer this question, but given their R&D use at the moment, have you done any side-by-side comparison with other commercially available microglia?
Our collaborators have done some comparisons with commercial cells, but I don't think I can disclose the information on those, I'm afraid.
Jenny?
How many different iPSC lines have you got?
So for the microglia, we have one that is already released, and we have one that is close to the end of development. So we'll have 2 iPSC backgrounds. This one is male, and it's fibroblast, and the other background that will come up would be from female donor.
Could I just follow up from that then? Can you tell us about the genetic background? Is the polygenic risk for Alzheimer's and other diseases known, or ApoE status?
For these, they are healthy, but we are generating, we've got 2 disease models that we'll be generating isogenic to this background and most likely to the other background as well. I don't know if I can disclose them yet, but I can, we could probably get in touch and let you know which ones they are.
Thanks.
Any more questions? If not, I'd like to thank Camilla and thank all the speakers this afternoon.